DNA replication or DNA synthesis. This process takes place in the cytoplasm of prokaryotes and inside the nucleoplasm of eukaryotes. So the purpose of this process is to produce duplicate copies of DNA needed for cell division and duplicates the genetic information contained in each DNA. The duplicate copies are all passed on to the daughter cells during mitosis cell division. Here are the enzymes that are vital in the replication process. Helix destabilizing protein, ligase, helix unwinding protein, gyrase, DNA polymerase III, RNA polymerase, and the DNA polymerase I. Here are the codons and the uracil. The process of DNA replications begin when helix unwinding protein splits the double-stranded DNA molecule by breaking the hydrogen bonds between nitrogen bases. As the HUP start to break the bonds, the gyrase enters. It relaxes the twisting tension in the DNA molecule due to the unwinding process. As the HUP continue to break the hydrogen bonds, the HDP enters. It binds to the open DNA strand to keep it from re-annealing.
When the HUP reaches the end, the RNA polymerase enters, and RNA polymerase enzymes synthesize short segments of RNA that serve as primers for the elongation of the complementary strand. The elongation of the transcript begins when the DNA polymerase III adds DNA nucleotides at the hydroxyl end of the RNA primers. At this point, the DNA polymerase I proofreads the new strand and replaces the RNA primers with DNA nucleotides, replacing the uracil with thymine. As DNA polymerase I replaces the RNA primers with DNA nucleotides, it leaves a gap in the sugar phosphate backbone, and the ligase molecules seal the gaps in the newly synthesized DNA strand.